on this computer. See, for example, this has not been recording, this stupid jerk. Okay, so anyway, there's always can be some hiccups that can happen with recording, like just now, how this for some reason wasn't recording. But please make sure to check in through Blackboard with these slides to know what's going on. Rewatch these lectures, and if you can, please be here, because that is sort of the safest way to tune in. Audio should be muted for the exam. That's absolutely correct. Your video should be on, your audio should be muted, and please use the text feature for asking your question. If for some reason the text blows up and the only way to ask a question is through audio, you are welcome to do that. Okay. I'm gonna also give you a sneak peek, basically, of how we're going to deal with exam next time. So basically what's going to happen for our next class period, I thought I had a slide for this, but next class period, right at the start of class, I'm going to launch emails. So I'm going to do one email blast through Blackboard, which is going to give you a practice, like, fake exam link. This will take you to a Google exam link. That is not your exam. You're not taking an exam on Thursday. This is all just practice. I'm going to give you that link right at the start of class. Then I'm going to give it to you through email. And then what's going to happen, basically, is you will click on that link, and that will allow you to access practice exam. You're going to throw on your video just so I know that that's working okay, and then you're going to submit this little fake exam just to make sure that you know how this is working. You have the tools that you need to do this. If you are one of my very small handful of students that has been taking exams through the Student Success Center, you will get an email Blackboard blast from me, but I'm going to hand email those folks with a separate link. And then technically in this case, it's going to be the same link, but ordinarily it'll be a separate link. And you will click on that email. If you don't get two emails from me, then we have a problem and you need to let me know. You'll click on that link, you will do the same thing. The only difference between your test and everyone else's test is the amount of time that I'm keeping that exam open. No other changes. You should already see sheets up in Blackboard that are your front page that gives you all the equations. That's up for the next two exams. You have access, you can go check that out right now if you want. Well, maybe not right now, but anyway, you already have access to that. So again, on Thursday, I'm going to send around a link. This link is to a practice Google exam. You're going to open it up. You're going to fill in your answers. You're going to submit your exam. You're going to show me your Zoom video. We're going to make sure everything looks cool. At that point, if you're like, holy crap, I didn't get an email from Dr. Soul, or if you took your exam from the Student Success Center and you're saying, holy crap, I didn't get 2 emails from Dr. Soul, you're going to contact me right then, and we're going to figure out what on earth has happened. I have another slide somewhere that goes into all this in detail. I should be up right now, but I don't know where it's gone. Does anyone have questions for me that they want to pop in chat right now about how we're going to do this on Thursday? This practice exam, not real exam, practice, practice, practice. Will you have access to the recorded lectures from last week? You mean this, Julia, in the context of Blackboard? I can't remember if last week we were at home or not. Okay, yeah. So somebody had emailed me at some point saying that they couldn't have access to lecture capture over the weekend. I'm not sure if that has been resolved, but in theory, all the lecture capture stuff should still be there. In terms of the last couple lectures that I've been doing through Zoom, what I've been doing is I've been making Blackboard announcements where I give you links so that you can go to my unlisted posting of lectures on YouTube channel. I'm not making them public just because a lot of you are showing yourselves and showing your names and showing your pets, and I don't want to harm your privacy in that because I'm not sure what all is being shared. But anyway, you should have unlisted access for basically every lecture we've had so far, and that is as a Blackboard announcement. If you're having a hard time doing that, please put the YouTube channel link in Blackboard. It's only in the emails, not in the other page. Yes, I will absolutely do that. Let me make a note to myself to do that. If I've done something and it wasn't what you meant, Laura, please email me and say that wasn't what I meant. But I'm going to do link to channel, And then I'm going to go ahead and just make a multiple links to all lectures so far. And I will do that after this lecture. I'll do that after this lecture converts and uploads. So it usually takes an hour or so after this lecture. So by today around 4-ish, you should have all of those links up on Blackboard, and that should be great. Any other questions about this practice exam, about things? Okay. Don't be shy about jumping in and stopping me. I just want to make sure we're confident about all of this. One super annoying thing that I want, well, not annoying, but annoying for you all, the link for the exam is separate from the Zoom link. That's exactly right. So you're going to get a link from me that's just going to be some Google Sheet link, essentially. It will not count as any points whatsoever. This is just simply to test everything is working okay, because I want to alleviate as much test anxiety as absolutely possible. I know technology is kind of a headache, and so I just want to make sure everybody feels good going into this exam. Just practice, just for fun, just to make sure everything's working. The link for the exam is separate from the Zoom link, so you will click on the Zoom link to join the class, and then you will click on this link to access the Practice Baby fake exam. The fake exam is like 2 questions. The main exam, I think, is something like 46 questions. It's a hair longer than normal. Basically, how I've set this up is you know, I'm expecting that you will have notes and books and all this stuff kind of at your disposal. So I've made a few more calculation questions than normal and more like conceptual things as normal. What this means is, and I've also made it a little bit longer, it's possible that folks may not do as well as they are normally doing and will curve just like normal, and it will be fine, because I know there's a lot going on. What my goal here is if you have studied well, you're able to do a lot of this exam, or ideally all of this exam, without using any aids. My expectation is that you're not using any aids to do this exam. The reality is, you know, things are complicated, and so you know, I'm not sure what people will actually do in reality, and I don't want to make a situation where it's fair for some and not for others. So I'm having the expectation that it's possible that you're going to have notes and things out. So you know, I realize that can happen, and that could be fine if we all sort of do it. I just want everybody to be on the same page. But the catch to that is, if you're spending your whole time looking through your notes, looking through the textbook, looking through things to find answers, you're not going to finish the exam. So what my goal is for you to do is to study like it's a normal test. Study just like it's a normal test. To the degree, you know, that you're normally studying and more, right? You know, study really hard for this, and then go through this exam and answer everything, ideally without using any help. And then you get to the things you couldn't answer. Okay, go and look into your help, look into your texts and whatnot, right? And try and answer those questions using those materials. The expectation is, if you use your notes and your book and everything for every single question, there is no way you're going to finish this exam in time. So that's the way I'm trying to sort of get everybody on the same page. The practice exams on Blackboard are not going to be altered. You just need to go through chapter, so exam 3, and look for all of the carbohydrate questions. That's the only thing that's basically being added to this. I'm not going to go through and combine those exams, because really I'm not wanting you to spend so much time on those exams anyway. You can have blank sheets of paper just to work out the problems, absolutely, and please do so, because I think it would not be ideal if you didn't. You're going to need to work those problems out just like any other time. Other questions about the exam and things like this? Okay, so the thing that I wanted to mention today, our dean, who's doing really a nice job in sort of helping everybody navigate this process, our dean is calling a college-wide faculty and staff meeting on Tuesdays at 2 o'clock, or I'm sorry, Thursday at 2 o'clock, which of course is our office hour, our second office hour this week. As a result, I'm going to have to cancel this Thursday's office hour because it's important I go to this meeting. I'm very sorry about that. Instead, I'm going to give you a makeup office hour. That will be Friday from 2 to 3. You'll use the same link that you used for Thursday. So just click on that Thursday office hour link, and please join me on Friday. I'm sorry for that change. It's for this week only. Do I have a suggested strategy for taking the exam since the exam is longer? Yes, that's exactly it. Just make sure you study like a regular exam. I want you to go through everything and answer what you can, the low-hanging fruit, just like always. Get as much of that exam done as you can for all of the easier questions that you can handle. Office hours on Friday, it's on this slide here, 2 to 3 p.m. Hopefully you can see my slide that I'm showing. Let me know if you cannot see my slide. So 2 to 3 p.m. Friday. I forgot my train of thought. Yes, so please go through that exam, get all the low-hanging fruit answered. It's not terribly much longer than your other exam. It's only about maybe 5 questions longer or so. But this is sort of the balance between not wanting to make it so long and so hard that I freak out everybody, because that's not what I want to do. What I want to do is just make it where I'm really testing your knowledge, where I'm acknowledging that you have access to materials for this to do so. Yes, Pacific time. Where you have access to materials to do this, which is a little bit different. So I'm just trying to keep this exam fair. Hopefully not adding additional stress, because I don't want to do that, but I understand that this is inherently a bit more stressful, but that's the rationale for doing that. I think next week is spring break, if I'm not mistaken. We do not have class or office hours during spring break. Yes. Any other questions regarding all the things? Sorry. No, Thursday. We still have Thursday. Thursday's when we do the practice test. Anything else? That was just like, I choked on my own spit. By the way, I'm not sick. Okay, anything else? Will I respond to emails during spring break? I will. I still am trying to get work done. So this spring break is always a great time for me to actually get a lot of work done. So I will be checking email. I may be a little slow to respond, but I will check emails and I will respond vaguely timely over spring break. Anything else? Problem set is today, because we're starting to learn new material next class period. So remember, check the updated syllabus and previous slides that we've been going through. So a lot of these things are things I think you have access to in the syllabus. So for respectful of everyone's time to get through material, I'm going to pause questions for now. If you have something new that can't be in the syllabus, please feel free to put it in the chat box. Okay, I think there's a chance we might have a little bit of extra time today. So yes, carbohydrates are on this exam. So exam 2 is gonna be chapters 4 through 8. That's 4 through 8. Okay, so if we have time at the end of class, let's do a Q&A, kind of like office hours, so we can cover some things. First though, we need to talk a little bit about what we didn't get time to get to last week, and that is the last bit of carbohydrates. Okay, so we started to talk about polysaccharides. So polysaccharides obviously are just like what they sound. They're a collection of monosaccharides that are linked together, linked together, linked together. And we spent a lot of time talking about polysaccharides in the context of, and really disaccharides in particular, their linkages, whether they're head-to-head and they're non-reducing. So they're 1 to 2 linkage, right? If they're head-to-head, no reducing, no anomeric carbons are free. And we also talked about 1 to 4 linkages, right? Where they're head to tail linkages, there's an anomeric carbon free, it's a reducing sugar. You can link additional polysaccharides onto that anomeric carbon. Okay, but now we're going to talk about, in particular, some of these actual polysaccharides. So let's start with chitin. So chitin are basically all of these really tough things that you're seeing on a lot of these organisms. Some of these you might know is to be delicious. I don't know if any of you have started eating insects and stuff, right? I think I've had some cricket powder in a baked good once, and I didn't barf, so there's that. But I know I'm definitely into some of these other types of organisms for eating. But these are basically these really hard exoskeletons. Their linkage, we had time to talk about cellulose, but they're a little bit different from cellulose because it's not just straight up glucose. You do not need to be able to recognize the structure on these N-acetylglucosamines. You don't need to be able to recognize that. I'm just wanting you to know that they're a little bit different than glucose linkages that we see in cellulose. And these strands can either, kind of like when we were talking about beta strands, they can go together parallel or they can go together anti-parallel. So let's take a peek about what these things actually look like. So check this out. Basically what's happening here is we have some linkages. So remember, all right, here I go, I'm going in. All right. Do I also have like an undo? I do. Holy crap, look at me go. And I'm going to do a different color because, whoa, okay, here we go. All right, so basically we're going to try and find the anomeric carbon. So remember, we always find that oxygen first, and we know that the anomeric carbon is going to be the carbon that is next to that oxygen, always, right? So now we just got to figure out, we got a 50-50 chance now we got to figure out which is which. So if we have just a single monosaccharide, then it's always going to be the carbon that has an OH group on it, always. If it's linked, it's a little bit harder maybe to recognize what it is, but it's still kind of easy to figure out what's going on. So let's dive in here. All right, so we have this oxygen. Let's start at this carbon first. Okay, so in this carbon, we have a CH2OH, right? Which could mean that we just had a ketone, right? Rather than an aldehyde. But look at this other group. It's just a hydrogen. There is no OH group here. This cannot be our anomeric carbon. No way, no how. Let's check this one out. Okay, so we have an H here. And then, it's kind of hard to tell what has gone on here, right? There's an oxygen, but we don't really know where it came from. Basically, the fact that we did not have an OH group here means that we have great confidence that this is going to be our anomeric carbon. Now let's look at this other piece here. So again, here is that oxygen. So we're either talking about this one or this one. So let's again start here. We have a CH2OH and we have an H. We do not have an OH here, right? So we're going to nix that one. This must mean this is our anomeric carbon. And so this used to be this one's OH, essentially. This is our new stereocenter. How does everybody feel about recognizing that? Hopefully you would feel good about recognizing this as a 1,4 linkage. Now, a lot of you are kind of panicking a little bit of like, oh, I got to count the carbons, holy crap. And no, not really. Basically, we're almost always talking about a 1,2, a 1,4, or a 1,6. And your 1 to 2 is always going to be the head-to-head, so anomeric to anomeric carbon. So if you know that you have a head-to-head linkage, it's going to be a 1,2. 1,6 is super wacky looking. I'll show you that in a second. And so 1,4 is all that's left. So it's really not so bad in terms of counting carbons. Okay, so these are not don't have intense linkages, so these can actually stack together really well. I'll show you some alternate versions where they actually don't stack together really well because they're highly branched. This means that you're really maximizing your hydrogen bonds here, which is pretty cool. Okay, so let's talk a little bit about some of these options that are outside of structural support. So these are actually energy support. So we talked about cellulose and chitin as examples of polysaccharides that are going to be really important for giving us structural support. This is energy. So we're going to talk about two kinds here. We're going to talk about glycogen, which is what you and I like to do in terms of storing up energy. And we're going to talk about plants, starch. So these are what plants are super into. Mobilizable, I'm pretty sure that's spelled right. So whatever, Google. But anyway, these are basically ways that we can store and access relatively quickly energy whenever we need it. In both cases, we have glucose that are present. And the difference, I'll show you between a little bit, but these actually have some of these interesting linkages, some of these 1,6 linkages. And I'll show you what that means structurally, but the moral of the story, the take-home message is that these become highly branched. Okay, so let's take a look at starch. Here's the difference, right? Check this out. These are basically, in this little part here, this is basically showing that if you don't have a lot of branching, you can really stack these together. If you're wanting basically a lot of good support, structural support, you want something that can be stacked together effectively like this. If you want to have really a lot of options for storing food, right, in a sense, this branching is actually really helpful to help you do that, because it allows you basically a lot of adding points basically to put carbons onto, right, or to put monomers onto. If you have a lot of these branch points, you have a lot of open-ended things where you can add even more carbons to, or monosaccharides to. Okay, so basically in the case of starch, we have a couple of different linkages here. We have 1,4 and we have 1,6. These are present in amylose and amylopectin. Again, this year we're not worrying about structures, but it's basically these 1,6s that are giving us a lot of these types of branch points often. And you can start to see these branch points in this little part right here. You can see how sort of disorganized looking this is essentially. Okay, so these are the differences of how these things are actually being linked. You can see that this is a 1,4 linkage, right? In this particular case here, you should be able to recognize that as a 1,4. But look, this is kind of funky, right? This 1,6, where we recognize this is an anomeric carbon, this is an anomeric carbon, but we're actually involving that little tail for once in our lives, which is kind of interesting too. Okay, so this basically is kind of showing us sort of a broad view of what happens. This amylose kind of going in nice little chains here as they have these 1,4, 1,4, 1,4 linkages. Okay, looks like Courtney's okay. And then you have this branching basically that's happening in amylopectin. And this branching is happening pretty often. So every 20 to 30 residues basically with a, well, residue is not a good word for that, but every 20 to 30 carbohydrate or monosaccharides essentially is a better way to describe that. Okay. And then this is actually kind of interesting, right? Where, you know, I don't know if any of you have basically tried to chew on some, you know, cellulose on the side of the road. Probably not, hopefully not, but wouldn't go super well for you. Your GI tract would get cleared out pretty good. So I guess there's some, you know, use in that. But basically, these types of linkages are really important in the context of whether or not we can actually break things down. And so in the case of starch, which we can actually digest in our stomachs, they have 1,4 linkage, but the key thing here is an alpha, right? And so that's showing you basically this alpha, which is giving you not a branch, but a bend, right? So your 1,6 is giving you some branching. Your alpha linkages are giving you some bending. And so we actually can handle these types of linkages pretty readily. We can chew them up. And so if you eat some starch, which I don't know about you, but I think is highly delicious, how can you not like French fries? You can actually break these down pretty effectively for a tasty meal. In the case of cellulose, not so delicious, super fibery. So if you go and gnaw on a tree, what's going to happen to you is basically you're not going to be able to digest your tree very well. And that's because of these beta 1,4 linkages. So we don't have enzymes in us that are able to chew these up, break them down. And so we end up just having a lot of fiber that's passing through us undigested. And there's a ton of interest in trying to figure out how to effectively actually break down cellulose as a food source, because there's a lot of cellulose around. The difference between the alpha and the beta linkages, we actually kind of, we really focused on alpha and beta in the context of recognizing that on a single monosaccharide, because it's a little bit easier to see. So remember, it's just simply referring to, let's just go to one of these, for example, or let's go to this. It shows us a little bit better. So remember, this was in the context of like, where does the OH basically on the anomeric carbon fall in relationship to where that CH2OH is. And so if these things are basically on the same side, that's going to be a beta. If the OH on the anomeric carbon and the CH2OH on the opposite side, that's going to be an alpha. an alpha linkage. So remember, alpha opposite, your two vowels, and then beta is going to be the same side. It's can be a little bit complicated. This is an example of a beta 1,4 linkage, and maybe that's easy to see because it's on the same side. But sometimes it can get a little bit confusing because you're not sure and how they've drawn the structure. It's not always clear. Like for example, structures that are kind of like this, you know, it's a little bit kind of hard to tell what's going on maybe. So I'm going to have you recognize alpha and beta in the context of a monosaccharide. Does that make sense, Laura? Okay, so in the context of glycogen, this is really a lot like starch. And it's really just kind of this difference in linkages and things like this. So this is how we like to store our excess potatoes and other types of things, other starches and such. And so we basically, our liver is really into this. We use this as a storage molecule highly in the liver, a little bit in other places too. And the only difference really that's happening here is between this and starch is the degree of branching. So we already talked about, we talked about the bending that happens from the alpha and the beta, but we also have the 1,6 linkages that are giving us branching. Right. And so the reason why this is so valuable is because if you're always going, you know, let's say 1 to 4, 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 that means for every 1 to 4 grouping, right, you only have a single reducing agent free. So let's take a quick peek at this, right? So here is our anomeric carbon, and here is our anomeric carbon. Well, look, this one's busy, right? It can't be doing stuff right now because it's already involved in a linkage. We only have this one free. And if it is further linked, then that one's not free either. We have to go all the way to the end of the chain. But check this out. If sometimes you're up in here, right, where you're actually, you know, up in this section doing a 1 to 6 linkage, that means, hey, look, I actually have this one free now. So that means that you can have make building blocks, right, with a lot of these types of linkages. And then, you know, you could have extension out the back here. You just have a lot more sort of reducing ends that are available. Does that make sense? Hopefully so. Okay, let's give this a whirl. Do you remember about epimers? Hopefully you remember about epimers. Okay, so hopefully, so starch has 1 to 6, but yeah, exactly. So both of them have these, has the branch points, the 1 to 6 branching, but glycogen just has a lot more than starch. Okay, so remember when we were talking about epimers, we were talking about molecules that are going to have stereocenters, sure, but but epimers are a very special class where they only have a change in stereochemistry at a single position, So in this case, we have a stereocenter, a stereocenter, a stereocenter, a stereocenter, right? This is not a stereocenter. This is not a stereocenter. All right, this is the same, right? Same stereochemistry. Same stereochemistry. Different stereochemistry. Same stereochemistry. So that's good news. Let's check out what went wrong in some of these others. Look at this. Mirror image, mirror image, mirror image, mirror image. Every single one of these is flipped. All right, let's see what's going on here. So mirror image, mirror image, mirror, no, sorry, mirror, so mirror image, same, right? Mirror image, and then same. So we actually have two things here that are the same, but we have two that are different. So this has one stereocenter difference too many. Okay, why cellulose can't have 1 to 6? So basically, so that was, I can't pull up that slide without too much drama. So basically in our previous lecture, we were talking a little bit, whenever we were talking about cellulose, this is basically our previous lecture. And so what we were discussing in the context of that was also in the context of some types of linkages and some types of branching as well, right? And so in the case of the starch, it's really just simply a number of the different types that we actually have that's present, right? So, or the number rather. So in the case of starch, we actually have a lot fewer of these types of linkages. In the case of, no, that wasn't the question. Why cellulose can't have 1 to 6? Okay, yeah, so sorry, I misunderstood. Okay, so cellulose and chitin, these are both of the other two kinds of storage molecules, well, types of polysaccharides. These are the storage class, not the energy class. In the energy class, you want to have a lot of branching points so you can be stacking a lot of things, have a lot of reducing ends free. In the case of starch, or I'm sorry, in the case of cellulose, in the case of chitin, you want it to be sort of flat and squished together and strong, right? So you don't want branching points. You can't stack branches well, right? But if you have, you know, just these straight line, these nice linear chains of polysaccharides, those stack really well. So that's why we don't have branch points in our structural types of polysaccharides. So if this type of, does that make sense? Hopefully, Yasser. So this was the exam we're just looking for. Yes, exactly. So we're just looking for which of these epimers has a single change in a chiral center. Okay, so try this, please. Okay, so remember what we're wanting to do here is first, always, always, always find the oxygen, right? That way we know it's either going to be this one or it's going to be this one. D, C, and B, you should not even be thinking about, right? It has to be the carbon that's adjacent to that oxygen. So that takes out a lot of them out of the running. Our next rule, if it's a single monosaccharide, it's actually pretty easy. It needs to have an OH, right? It's in a different, it's flipped in the opposite direction than what you're used to seeing, right? But check this out. It's got an H and it's got a CH2OH. No OH, cannot be A. In this case, we have an OH. This used to be a ketone, not an aldo, so we still also have a CH2H hanging off here. This is the only one with the OH. That means we're looking for E as this answer here. Hopefully that makes sense. Okay, so any questions about some carbohydrates before we go on to today's lecture? All right, I'm imagining all of you just patting yourselves on the back being like, damn, I get these carbohydrates. Okay, so let's talk a little bit about therapeutic peptides for a second, because we're going to use this as a means to, yes, damn, you do. We're going to use these as a means to basically really familiarize yourselves with how amino acids are shaped and make sure you're familiar with their properties. Okay, I don't know if any of you are familiar with peptides as a therapeutic agent. I don't know if you have a loved one that ever takes insulin. That's a great example. There are some immunosuppressing types of drugs. Sometimes there's some anti-cancer drugs that are peptides. But biologics, maybe you heard them as biologics. These are really really making huge waves right now, and actually have for decades if we're talking about insulin. But these are really effective as drugs. They're kind of hard to deliver. They're usually delivered through IV, just because sometimes our body kind of chews them up. They never get to the place where they need to be if they have to go through the stomach. But anyway, they're super popular. They're pretty awesome. These are just sort of a series of different things where we might find peptides. We have also natural peptides too, right, that we're using as signaling molecules, but there's all kinds of cool stuff that's going on. Okay, so the thing that I want you to appreciate is that anytime a peptide is interacting with its protein, or even a protein is interacting with a protein, or whatever, we're always taking advantage of these little types of interactions that we've been talking about as soon as we started talking about proteins, right? Ionic interactions or salt bridges, hydrogen bonds, hydrophobic effects, van der Waals, disulfide bonds. All of these things are going to be really important to make sure that proteins interact well with their proteins or peptides interact well with their proteins, whatever. So you should be really familiar with basically the types of information, the types of interactions that are happening among these. I just relisted this here just for your own review. But let's talk about what you might be thinking about if you were to design a therapeutic that's going to bind really well. So here's what's going on here. So check this out. So we have some protein, right? Some protein that maybe you and I have in us that we're really interested in designing a therapeutic peptide to recognize. And you can see that I've drawn, you know, two-dimensional, sure, but I've drawn a little bit of shape associated with this, where we have, you know, basically a big distance here, right? And then sort of smaller distances and all of these other locations. And basically, we're trying to design a peptide that basically binds flat, you know, sort of in a straight line like this, just for ease. Where you could imagine that you're going to need a longer peptide or a longer amino acid here, right? But maybe a shorter amino acid here. And so what I'm wanting you to do, basically, is to figure out what should be the best amino acids, and there's not one answer, there are many answers. What are the best amino acids that you should put in opposite each of these residues, right? I'm going to clear out this nonsense because I'm not even being useful anymore. But basically, you're going to have an amino acid opposite each one of these things. And so that's going to be your first test, is designing a peptide that's going to bind just like this. So that's basically what's going on here, essentially, right? You're going to be familiar with the types of reactions that are involved. You don't have to have them all represented. It's okay. You just try and make a really good, strong reaction. It will probably happen naturally that you'll have all represented. I then want you to figure out what your overall charge is. This is going to be at physiological pH because this is in humans. So this is the overall charge of this sequence here. And then I want you to figure out basically what is the charge of your peptide, right? So by charge of this guy. And then we're going to talk about if you need this to bind to a column, how are we going to do that, right? So you should have access to your structures of amino acids. You should already be pretty familiar with them anyway. But I'm going to give you, this is going to be super awkward for me, so I'm just going to stay entertained, to be patient. I'm going to give you a good 10 minutes to work through this. I'm going to be monitoring the chat in case it's not clear what to do. And I'm going to clear out my doodlings because that's not helpful. So let's take about 10 minutes to work through this. you're having a hard time getting started, or if you're having any confusion, don't be shy about reaching out for help. I want to make sure that you're using this time to work on this problem, not to just to be totally lost. Yeah, exactly. Okay. Um, okay, thank you. It's really hard to know what's going on. I don't want you sitting in like uncomfortable silence, not knowing what the hell's going on. Okay, so what I'm really wanting you to do, if nothing else, please just start at the very beginning part. Um, Let me get you started here. Let me do these first two for you. So we have a phenylalanine, right? What is in? Anyone remember? Hopefully we said asparagine, right? Okay, so we're not actually looking for bonds necessarily, right? But what kind of interactions would phenylalanine like to make? Would it want to make ionic interactions, disulfide bond interactions, hydrogen bonds? Nope, it's exactly right. We're going to want hydrophobic interactions. Okay, so not only that, check this out. We're designing this sort of flat peptide like this, right? We don't have a ton of space here, right? If I were you, I would put an alanine or a glycine right here because it's small and it's hydrophobic. All right, let's do this second one now. For this next one, we've got that asparagine here, right? Remember, this is a polar non-charged residue. And we have a little more space to work with, right? It's not as big as phenylalanine, although it's kind of big in its own way, but we have a little more space to work with. So I'm going to want something that's, you know, a little bit bigger, and certainly going to be polar, right? I could put threonine there. I could put a tyrosine there. Heck, I could put, you know, a D or an E. I could put either of those, aspartic or glutamic acid. I could put another N opposite that if I wanted to. So a lot of these are going to be correct answers. You're going to have a lot of flexibility what's happening here. So go through each of these amino acids and just make sure that you're having appropriate pairings at each site. Once you do that, let's figure out what the charge of this thing is, figure out the charge of your own peptide, and figure out how we're going to basically purify this thing. If you don't get that far, that's okay. I'm going to give you another, let's say 4 minutes, at least have all of the amino acid pairings for those interactions. Q would be okay too. All right, so I'll give you another 4 minutes, get as far as you can. Yeah, hopefully that helped for a couple of you that were a little nervous. Yep, so charge at pH 7, that's right. We'll do everything in terms of pH 7 here. So basically, you know, when you're trying to make the call between should I do a polar uncharged or a polar charged, you know, a charged an ionic interaction is going to be stronger, you know, than just sort of a vague nonpolar, you know, type of spiel. Although on an exam, I'm going to make it pretty obvious what a bad call is. If I'm going to have, let's say, you know, if I'm going to have a leucine opposite an aspartic acid, you're going to recognize that as a bad call, right? So if you have, you know, an in, um, you know, you can choose an E, you could choose a K, right? You know, if you have a positively charged lysine there, you probably wouldn't put it in another lysine opposite it because you don't want two positive charges. It might be better to put an E opposite that, so you have a positive and a negative charge, but you could also put a glutamic acid and just take advantage of a polar interaction. So there are many right answers here. Okay, so I'm gonna go ahead and have your time up here. Basically, This is just kind of the slide I'd normally show you just so that you'd have access to structures, but you probably already did. There are many, 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 many solutions that are possible here. This is just one single example of one right answer. A couple of things I want to highlight here. I went with a cysteine-cysteine, right? I want that disulfide bond there. Rationale for this is hydrophobic and small. This is hydrophilic and a little bigger, right? This A all the way at the well, I went for something really big, right? And hydrophobic here. I went with an ionic interaction here because I have, you know, a possibility of a charge. So I wanted to take advantage of that. This is just simply hydrogen bond, another example of a hydrogen bond. I could have gone with, you know, a D or an E here and had a ionic interaction, but there are a lot of different options here. Can you all give me a shout out if this really doesn't make sense at all to you? Okay, so now we're basically kind of dealing with the situation is we need to figure out what our overall, okay. So basically, what I'm wanting you to do here is to know the properties of the given protein, right? Of each amino acid of the given protein. You should recognize that lysine is going to, at a pH of 7, it's a polar charged molecule. Cysteine is special. It can actually make disulfide bonds, right? So we can actually have, do it more, we'll do it in this version, right? Disulfide bonds. Threonine, that's a polar molecule. Remember, it's got an OH group here, right? So this is a great example to do a hydrogen bond. This is actually a polar charged, right? This aspartic acid. This alanine, basically just a little methyl group here, right? In the case of asparagine here, we've got basically some nitrogens that we get to work with here. We also have an oxygen out here. In the case of phenylalanine, remember, this is just simply a ring, right? An aromatic ring. And so what we did is we understood all of these different properties of each of these amino acids, and we paired them with a like interaction, essentially. So we're following the curve in the context of how much distance do we need to span to make this interaction, right? This is basically our longest distance we have to span, right? This is a pretty short distance. This is a pretty short distance, right? This is a really long distance. So in this first example here, I need something that's hydrophobic and pretty small, right? Because this is kind of at the upper part of this curve here, and it's also a really big amino acid in and of itself. So that's why I went with an alanine. Could have gone with the glycine, just as good, right? And I could have done an ionic bond here. I just didn't, you know, but totally, you could totally put aspartic acid, glutamic acid here. That's a-okay. Okay, so in this example here, we need to take advantage of hydrogen bonds and a little bit bigger, right? So I didn't go with a glycine or alanine here. I went with something a little bit bigger. I needed something really big here that's also hydrophobic. That's why I went with a tryptophan. I did go with an ionic bond here. As I mentioned, I could have done that here too, but I'm just showing you that you can do a variety of things. But I wanted to give you an example of something that would not be okay for opposite of threonine. You know, if I put, let's say, for example, an isoleucine, right? That would be a really bad call because this is a non-polar residue and threonine is a polar residue. So I need to go with something that's polar where I can make H bonds. You could also do something polar here opposite cysteine, but it's really great to take advantage of a covalent bond here. And again, in this particular case, I just did a hydrogen bond, but I also could have done an ionic interaction. That would have been fine. Does that help? Those that took one for the team and admitted that this is a little confusing. Are we feeling better about this now? Okay. All right, good. Okay, so let's say that we have this particular peptide, right? What is going to be our charge that's present at pH 1? I'm going to give you a little bit of time to go over this. hide the slides. Oh, gotcha. Thank you. Well, actually, I can't on my iPad. I'm sorry. I can't on my iPad. Just please don't look. Or let me see. I'll see if I can change the layout, but I don't think I can. I'm sorry, I can't. Please just don't look. All right, would this get easier by practicing these kinds of problems? So you will have one problem on the exam like this, one problem. So, you know, don't spend all of your time on it. It's just one problem. But I would want you to do something like this on the exam. So all this is making you appreciate is size and property of amino acids. That's it. That's all that this is about. It'll be a little easier on the exam because I'll be giving you lists of possible peptides. And if you see that I did a polar and hydrophobic pairing or a hydrophobic and polar pairing, you know that's a bad call, right? So it'll be a little easier on the exam. Does that make you a little less apprehensive? Nope, it won't. The essence of this will be present on the exam. So let's actually go back. You're right. We didn't actually cover the rest of this. So let's just go with this particular sequence. Let me try and see if I can erase some of this nonsense on here. Let's see. This looks like an eraser. Yeah. Okay. So we'll come back to that multiple choice. But you're right. Let's actually go back and look at charge. Okay, so here we go. So what we're trying to do is figure out our overall charge that's present on this particular molecule. All right, so these are basically getting me to remember that I need to worry about the beginning and the end. At a pH 7, right, I'm going to have an NH3 plus at the beginning, and I'm going to have a COO minus at the end. That's telling me about these two parts of this chart that you're going to have on that front page of that exam, right? And then I'm just looking for, do I have any of these guys present, right? So let's look down here. So non-ionizable, not ionizable, not ionizable, boom, ionizable, not ionizable, right? It's only tyrosine, not, well, actually this one's ionizable and this one's ionizable, right? So we have to be worrying about these particular things, plus the beginning and the end. Okay, so according to this chart for D, we're dealing with a pKa that's around 4. For cysteine, we're dealing with a pKa that's around 8. And for lysine, we're dealing with a pKa that's around 10, right? So what we're dealing with as a pH equals 7, you're going to be asking yourself, am I above or below that pKa? So in this case, 7 is above 4. That means I'm deprotonated. In this case, this is my form. I got a charge of -1. The case of cysteine, right? In this particular case, I've got a pKa of 8, right? My pH is below my pKa. That means I'm protonated. That means I have this, right? No charge. Technically, I will be ultimately involved in a disulfide bond, but right, doesn't matter regardless, I have no charge in this case. Here I've got a pKa of around 10. My pH is below that. That's going to be protonated. That means I'm going to be this form of lysine, right? So that means I've got a +1, -1, neutral, -1, +1. How many charged groups? 1, 2, 3, 4 charged groups. What's our overall charge? +1, -1, these cancel out. 0, +1, -1. Holy crap, look, I actually have 0 charge here, right? Make sense? pK chart on the clicker question slide. I can't because I'm working off my iPad and I can't copy and paste them over. I'm really sorry. But I'll tell you on the next slide, I'll just tell you what you need basically, but I can't do that easy here if I'm working on the iPad, unfortunately. Okay. All right, so does that make sense for finding the charge of the protein? Okay, now let's think about finding the charge of the for the peptide that we designed, right? So I'm just going to clear this out a little bit here. Okay, so now in this case, what I'm dealing with is, again, a beginning and an end. And I don't have to worry about this, right? I got to worry about this. Don't got to worry about this. Got to worry about that. Don't got to worry about this. Got to worry about that. Don't got to worry about this, right? Okay, so we're dealing with pKa. Check this out. 10.5. Arginine, let's see, where's my arginine? 12.5. Cysteine, 8.4, right? And then the beginning and the end, which is 2 for the end, 9 at the beginning, right? All right, so we got, again, a pH of 7. Okay, so what this means is that, again, NH3 plus over here, COO minus over here, right? Tyrosine, let's see what's happened at tyrosine. 10.9, that means my pH is below my pKa. That means I'm OH. That means I'm neutral, right? No charge there. Let's check out arginine. Arginine, where's that? Here it is, 12.5. pH is below that. That means I'm protonated. That means I've got some NH3 plus going on here essentially, right? So +1 charge here. There's that. Cysteine, same spiel as before. This is going to be in the SH form because I'm going to be protonated because 7's below 8. 0 charge here. And then I just got the beginning and the end, right? So how many charge groups? 1, 2, 3 charge groups. What's my overall charge? +1, +1, -1. So this particular peptide is an overall charge of +1. Okay, does that make sense? We're doing okay on this. Okay, so what this means is I want to actually purify this thing, and I want to purify this at a pH of 7. What kind of column do I need to make sure that my positively charged protein is going to bind? Well, I need this to be negatively charged right here, right? I need resin that has minus charge everywhere. Well, what kind of resin has minus charge? Cation exchange, that's right, cation exchange, because we're exchanging out the cation, right? So negatively charged resin for cation exchange. This would be really great in order to bind our positively charged protein. And then when we add salt to dislodge our protein, we're really interested in the sodium ion to help dislodge it. Does that make sense? Okay, so that's basically those types of questions will be on the exam. So figuring out what kind of peptide would be binding here, you know, figuring out overall charge, a lot of that's going to be on the exam. So make sure you're feeling really comfortable with that. Although, yes, you still need to be really familiar with these, with your amino acid properties and structures. I understand that you can readily have access to this. So if you're wanting to make sure that you just have a picture up that shows your structures of your amino acids, this year that's going to be fine. Be aware if you're taking pre-professional exams in the future, you still will need to memorize those, but you're going to want to be able to do this quickly. So if you know right off the bat what class glutamine belongs to, even if you don't know the structure, you know that it's polar uncharged, that's going to be really helpful. So I would prioritize memorizing my classes, right? Memorizing the 1-letter codes, and then, just go to notes for overall structures, probably in this case. That makes sense what we just did here. Okay, so what I'm going to do is I'm going to reload all of these lecture slides so that you have all the answers. We aren't going to get through all the questions here today, but I want you to have access to them. So let's just go ahead and give this a whirl, just because let's try really hard to get in 2 questions. So I'm going to give you a couple minutes to work on this. So if you can maybe write down this polypeptide, and then I will show you the previous slide that shows you the other stuff. Or I'll just keep flipping in between maybe. So hopefully we have that written down. I'm just going to periodically flip between the 2. Okay, let's work through this problem. Let's go through these and figure out which ones we have to worry about. We always, always, always have to worry about the beginning and the end, right? Threonine? Eh. Heck yeah, lysine. Heck yeah, aspartic acid. Heck yeah, glutamic acid. Asparagine? Nah. Leucine? Nah. Glycine? Nah. Alanine? Nope. Tryptophan? Nope. Glutamine? Nope. Serine? Nope. Isoleucine? Nope. So here's what I got to worry about. These things, right? Okay. So I think I might vaguely have this memorized. You can correct me if I'm wrong, but this is somewhere going to be around around 9 pKa. This is around 2 pKa. This is about 4. This is about 4. This is about, I don't know, 10-ish, 10.5, 11, somewhere in there. Okay. So we're dealing with a pH of 1 in this case, right? Okay, let's see what's happening here. In this case, our pH is below our pKa. That means we're protonated. That means we got this going on, right? Okay, so far so good. What about whenever we're dealing with lysine, right? Okay, well in this case, we're dealing with, again, another amine group, right? We're trying to figure out whether we're going to be charged or not. Our pH is well below our pKa. Means we're going to be charged here, right? So positive charge. Look, this is unusual. We actually have a pH below the pKa of these two carboxylic acids. That means, unusually, we'll actually be protonated here. So 0 charge on both of these. And then this is basically the same here. We have this equilibrium that we're dealing with, right? And that means at a pH of 1, we're going to favor this form. So again, an overall charge of 0. So how many charged groups? 1, 2. Overall charge plus 2. Why is glutamine not ionizable? Well, if you want the smartass answer, it's because it's not on your chart, but you probably want the science answer. But the structure of glutamine does not have a free, basically a free proton that can be freely exchanged, right? It's very similar to asparagine is in the same type of boat, right? Where we have a nitrogen, right? That we cannot pull off that a proton off of that nitrogen. We have an ketone group that we can't pull or add protons to either. So it's only as far as our amine containing amino acids, it's just lysine and glutamine. Does that help? Okay, so that should have gotten us that. So that's happy news. Let's just take these 2 more minutes and work on this real fast. So remember, only sugar on the left in terms of this linkage. Don't worry about what's going on over here. Hey, I figured out how to hide it from you. Sorry, I should have figured that out before. Sometimes I'm kind of dumb. Yes, I'll quickly go through the rest of these. I have another Zoom appointment at 2. So I'll we'll end right on time, and I'll quickly go through the rest of these off of this same Zoom talk. Okay, so here's basically what we have going on here. Anomeric carbon, right? So we have an oxygen, we have 1 carbon here, 1 carbon here, carbon here, carbon here. We're looking for an OH, no OH here, no OH here, right? Here's an OH, we got a free linkage. We know this is gonna be 1 to 4, right? So this is our anomeric carbon. We have a reducing sugar here. Okay, alpha and beta in terms of just this piece on the left here, right? In this particular case, we have the OH group on the same side as the CH2OH. That means it's a beta. This gives us a beta 1,4. Okay, I'm going to continue yammering about the rest of these to take me through 2 o'clock. Everybody, I'll catch you later, and I'll put this whole video online. I am not going to give you time to fill in so I can make sure I get this done by 2. Everybody take good care. I'll see you all on Thursday. Please remember the change in office hours to Friday. Okay, so just chatting with myself. Let's go through this. You're welcome to stay too if you want. If you're a glutton for punishment. Okay, let's go through each of these individually. We're looking for cooperativity in terms of oxygen transport. This first option, this is basically, we're looking for true answers essentially. So cooperativity is critical because this helps the protein release oxygen at low concentrations and bind in high oxygen concentrations. That's exactly it, right? Remember, we have this sigmoidal plot rather than hyperbolic. For myoglobin, we have a sigmoidal plot for hemoglobin. And this means that we stay low oxygen bound for high concentrations, right? And only until we get a much higher concentration of oxygen do then we start really binding oxygen in really serious levels. So that is absolutely true, this first one here. This protein becomes fully saturated in the lungs. Yep, that's the point. We want to load up oxygen in high concentrations. That's this part of the curve. We want to be releasing oxygen in low concentrations. That's this part of the curve. That's absolutely true. Sigmoidal character of the percent bound versus oxygen concentration graph displays a way to regulate or fine-tune affinity. That's exactly right, right? Check this out. Here's kind of dumb hemoglobin, or I'm sorry, dumb myoglobin, right? It basically dumps, you know, as soon as we get pretty, very low concentration of oxygen, it's only then that we start dumping our oxygen. But look, we're all the way out here. This is basically concentration of O2. We're all the way out here before even starting to think about binding oxygen. Then we bind it really effectively, right? So this is hinting at us all the neat kinds of regulation that are in play. Changes in quaternary structure modulate oxygen binding. Yes, we totally talked about this, where you have 4 subunits, and this little window in here kind of gets bigger or it gets smaller depending whether we're in the R or the T state. So it's absolutely changes that are felt in each of these subunits that help regulate oxygen binding. All of these are good. Okay, which of the following is true about anomeric carbons? All right, check this out. After a monosaccharide cyclizes, the carbon adjacent to the carbonyl carbon becomes chiral. Okay, so after basically what we're dealing with here, natural monosaccharide carbon adjacent to the carbonyl carbon becomes chiral. Oh, okay, I see what's going on here. Okay, so basically remember, it doesn't matter which one we have that we're dealing with here. Let's just say that we have an aldose, right? That's going to happen here. Let's just say we got this going on, right? Basically what's happening here is we're going to have some type of attack that's happening here, and then we end up, right? I'm just going to shorthand this a little bit, but we end up with something like this, right? And then this is going to go up here. What this is telling us is after this cyclizes, the carbon adjacent to the carbonyl carbon becomes chiral. So that's either this one down here, or, well, that's an oxygen, so that's not going to fly. This is not true because it is the carbon, the carbonyl carbon that becomes chiral, not the carbon adjacent. This one, this went from achiral and now it's chiral. So if this was eliminated, this would be true. Carbonyl carbon, it becomes chiral. Okay, in a glycosidic bond, if the anomeric carbon is free, this is a non-reducing sugar. That's opposite. This is a reducing sugar if the anomeric carbon is free. Can only form upon cyclization of ketoses, not aldoses. False, right? Both of these can work. It's just a matter of sometimes there's a CH2OH group down here, and sometimes there's just a hydrogen. So that's a-okay. Or I'm sorry, that's wrong rather. The key part of the carbohydrate is always involved in the linkage, at least for one of the key part of the carbohydrate that is always involved in the linkage, at least for one of the carbohydrates involved, the saccharide. Okay, so yes, this anomeric carbon is an absolutely key part, and it's has to be, in order for a linkage to occur, it has to be at least through one anomeric carbon, what is in the case in a 1,4, or actually 2 anomeric carbons head to head in the case of a 1,2. But it's always anomeric carbon that's involved in a carbohydrate linkage. So that is true. Okay, check this out. All right, what happened if you forgot to add SDS page? Proteins would, if you got to add SDS page when if you forgot to add SDS when you were running to your SDS page. Proteins would run according to their charge. Well, that is true. That is one of the things that will happen. They will also still go according to their shape, right? And then they're basically their overall size. That's what this one is saying here. Mass, just simply because that's what we're trying to do anytime we're running SDS page. Charge, because we forgot to add the SDS, we're not coding it in negative charge. And also shape, because we didn't denature it because we forgot to add the SDS page. So all of this is true. And then these are just not, they're true, but they need more information. This is the only complete answer. Okay. So dehydrogenase molar extinction coefficient. I actually don't have a calculator here with me handy, but I'll just take us through this. This is what we're looking for here. Absorbance is 0.58, so I'm going to write this out here. Molar extinction coefficient, so that's 11,350 millimolar -1 centimeter -1, right? And I'm going to multiply this by 1 centimeter path length, right? So my centimeters are going to cancel out. And then I'm solving for concentration. So I'm going to have 0.58 divided by 11,350 per millimolar, right? Which is going to give me some really tiny, tiny, tiny answer in millimolar. I'm going to multiply this by 1,000 to get me in micromolar, right? Which is what's being asked here. So hopefully the math works out okay on this. All right. Doing okay on time here. All right, you want to perform an anion exchange purification of a protein. Okay, so what the crap is anion exchange? Good question, team. Okay, so if it's an anion exchange, that means that we're going to be exchanging out anions. That must mean that we're going to have positively charged resin, right? So it's going to be looking like this. And whenever we want to elute that protein, we're going to be depending on the chloride ions in order to elute our protein. So we want to make sure that we've got negatively charged protein that's going to be binding to our positively charged resin. Our protein has a pI of 6.1. So below the pI, we're going to be protonated, which means we're going to have pushing positive charge. If we're above the pI, that means we're going to be deprotonated, which means we're going to be pushing a lot of negative charge, right? Which buffer would you choose for this experiment? Well, I want it to be negatively charged. That means I want to make sure that my pH is going to be greater than 6.1. Meh, meh, yay. So that's good. Okay, what else we got here? Which of the following is true? The phi angle. Oh, good. Phi, remember phi and psi angles? So remember, if we basically, we've got nitrogen, oops, right? Here's a C double bond O, right? Then we've got our alpha carbon here, right? And then this is going to continue on, continue on. So here's our peptide bond here. So remember, between our carbon and our carbon, it's going to be our psi bond here, right? And then in between our carbon and that nitrogen, that's going to be our phi bond here. This is the one with the R group, right? We do not have any rotation at the peptide bond. All right, the phi angle describes the torsion angle between the alpha carbon-carbon bond. Nope, that's the psi angle. No good. If 10% of the phi and psi angles are known, the peptide can be fully predicted, determined. No, we learned in crystallography, we need to know basically the x, y, and angle coordinates of every single thing. It's not enough just to have 10. That's not okay. The torsion angles expand the number of possible downstream additional torsion angles that the protein have. No, remember Leventhal's paradox? Basically, we can shut down some of these torsion angles by having some of these early folding events, so we're not exploring everything. So this would be decrease, and then it would be correct. Okay, Ramachandran plot shows strategies of how steric hindrance can be overcome. Nope, it just shows you this is okay, this is not okay. So it's not at all about showing you strategies to beat the system in physics. So we definitely want this one. Okay, and last one, Bohr effect. Holy crap, here we go. What's true about the Bohr effect? Remember, this was telling us about what's happening with protons, right? Protons binding. Also be familiar with 2,3-BPG. So review that too. All right, CO2 dissolved in blood increases binding affinity. Remember, if I have CO2 increased, that means our pH is increasing. That means there's more protons around. If we have really high CO2, that means we have low O2. That means we want to be releasing oxygen, right? Okay, so we know that in our head. Now let's go back and review what's going on here. CO2 dissolved in the blood increases oxygen binding affinity. Well, that doesn't make sense. If we have a lot of CO2 around, that means we have low O2. We want to be freeing the oxygen. We want to be decreasing, right? So that's not right. This is bad. All right, carbon dioxide dissolved in the blood causes a decrease in pH. That's exactly great. We just talked about this here. Increase in CO2 means more protons, which means higher concentration of protons, which basically is pH, right? A decrease in pH. An increase in protons causes heme to dissociate from the protein. This is a nonsense made-up answer. That doesn't happen. This is a prosthetic group that's actually covalently bound thanks to that histidine. And that, gang, is a wrap. I'm going to call it a day. If everyone is still, holy crap, a lot of you are still around. You know, I almost had this false sense of security that no one is here and I could just, you know, babble to myself. But now I realize that you're all here and listening. So, hey, everybody. Anyway, that's all I got. I will reload this extra thing. I got to get ready for a meeting at 2. I'm really sorry, Gina. You can shoot me an email or something like that. But I will see you all on Thursday, or save your question for Thursday. I will post in this lecture and work on uploading the PowerPoint, uploading the verbal talky stuff. Take care. Bye, everybody.